Our next speaker is Ignacio Ochoa Garrido. He is assistant professor at the University of Zaragoza and research scientist of the Applied Mechanics and Bioengineering Research Group of the Aragon Institute of Engineering Research and the CyberBBN. He is PI as well of the Group of Environmental Mechanics and Microfluidics of the Health Research Institute Aragon, and where he's coordinator of the program of new technologies and innovation applied to health. He's going to speak today about organ on chip applications for cancer research.
Thank you, Laura. And also, first of all, as all the speakers have said, I want to thank the organizers, the opportunity that they have provided us to talk a little bit about our technology, the organ on chip technology. I don't know if all of you know how much it costs to create a new drug. Actually, it's around $1 billion and more than 15 to 20 years. And one of the reasons why this costs so much in time and in money, it's because we have some fails, some false positives during the preclinical test. So why we have some of the positive falls during the preclinical test? One of the reasons is because, as you can see here, we are really different. Our brain cells are completely different to the kidney cells, from the heart cells, as we have mentioned before, and so on. And we treat them in vitro as if they were exactly the same. We put them in a 2D plastic plate in a very, let's say, non-friendly environment. We take them out from the neighbors, we take out the 3D structure, and we change their gradients and physical and mechanical stimuli. So what is the cell capable to do? Just to adapt or to die. So in this adaptation, what we have is that we are changing the behavior of the cell. So this cell is not going to be any longer a kidney cell or a lung cell or a brain cell. So we have been using 2D culture for a long period of time, and it has provided us a very, very useful results and very useful knowledge in the field. But now, in the recent future, we have just applied new strategies, as you could see here, 3D culture, as Pedro has mentioned before, not only in hydrogels, as also Manu has explained, but also just with the scaffolds, and the tissue engineering is providing new tools just to get closer to the physiological environment. For sure, we have also the in vivo models, where we have been for decades working on mouse and zebrafish models, and also with higher animals also. For sure, our gold standard is always the clinical trial with humans, but for sometimes it's not possible to do different studies on them because of ethical reasons. So, depending on what you are interested in, you have some advantages and problems with the different models. In 2D and 3D, the problem is that you don't reproduce, or the interaction between cells is not provided in the model. In the in vivo models, it's quite difficult to monitor what is going on. So this is also a problem. So that's why we have in the middle a new technology that is based on the microtechnologies created in the early 60s. And with these microfluidic models, we go to the cell scale, and we can provide channels and chambers in similar fashion as they are disposed or laid out in the body. So, Manu has explained a little bit about, and Manuel Doblare has also explained a little bit about technology, the organ-on-chip technology, the microfluidic technology for cell culture applications, and I'm just going to explain you just a few examples of why we have used this technology in cancer research. The first example that I want to mention to you is the brain cancer, is the glioblastoma. This brain cancer is a lethal brain cancer. There's no treatment, effective treatment. All the patients died after a median of 14 months. So there's no treatment, there's no hope for them. And that's why the only treatment approved is just the surgical resection, radiotherapy, and the treatment with the chemotherapy with temozolomide that is just getting resistant very soon in the patient. This tumor it's very specific. And if you want to create something, and if you want to mimic something, the first thing that you need to do is just to understand how it's in the body. So what we did is just to check the histology. I'm professor in histology in the medicine faculty, so let me to put in the importance of my subject. So as you can see here, let me show you a little bit about the histology of the patients. Here you have a blood vessel, but it's collapsed. This is something that is common in this disease, that you have a blood vessel collapse surrounded by a necrotic core. And after that, what we observe is a highly dense area, what we call pseudopalisade. Okay, if we are talking about cancer, all of us, we can think about the possibility of those cells are growing and proliferating faster than the others. This was an hypothesis. But there was another hypothesis that it was called, those cells are migrating from the area where there is no any longer blood vessels. So we have what we call a growth dichotomy. So we were in contact with some mathematicians. Always the multidisciplinarity is quite important, as you will see here. And they told us, okay, are you able to reproduce this structure in a microfluidic chip? And we said, okay, why not? Let's go. Let's go to do it schematically. You have a blood vessel, another blood vessels, a 3D culture with cells. And so you're telling me that you need to collapse one blood vessel. It can be done. And to perfuse the other one. Okay, we translate this structure, this physiological structure, to a microfluidic chip. And we did it in that way. What we observed is that when we were just perfusing both lateral channels, we were observing no changes at all in the cell shape or number. So this was interesting because it was just mimicking the healthy situation. So if we have all the blood vessels okay in our body, in our brain, so we will have no problems at all in the tumor progression on the pseudopalisade formation. But what happened when we collapse one of the two channels? We observed that there was a dramatic change in the density of the cells, promoting a cell migration toward the channel that it was perfused. So we were just, with this very simple experiment, microfluidic experiment, mimicking the formation of the pseudopalisade in the brain tumor. But one of the questions that arise during the experiments was, okay, who is triggering? Who is the responsible of this? Is the oxygen? Is the nutrients? Because one of the questions that is important to mention here is that our chips, they don't allow the oxygen or the gases to come over. There's no perfusion, there's no diffusion of the oxygen through the plastic. The nutrients and the oxygen comes only through the lateral channels, mimicking exactly what it happens in vivo in our body. So we said, okay, let's go to check what happens with the glucose. We put a gel without cells, and we perfuse a fluorescent glucose, and we observed that there was no gradient at all. The glucose was not uptaken by the cells. When we put the gel with cells, we observed that there was not gradient at all. There were some peaks due to the lack of, it was impossible for the light to come through the cells, so that was some dark points, as you can see here, but it's not because of the lack of glucose. What happened when we did exactly the same experiment, but instead of checking the glucose, checking what was happening with the oxygen? Here, what we could observe is that in the unrestricted conditions, there was no gradient at all in the whole chamber, so this was more or less exactly the same as glucose. But when we were just checking what was going on the thrombotic chip, we observed a very clear gradient formation of oxygen. So this is telling us that one of the main responsibles of the pseudopalisade formation is the lack of oxygen. That is very, very important just for new strategies for this tumor. Another question was, as Manu has mentioned before, is, okay, what is going on? How the cells migrate inside a chip? How the cells migrate better inside a body? So we know that the cells are in 3D, as I'm a 3D person, so, That's why we are always trying to mimic the reality, and we said, okay, let's go to check what is happening in a head and neck tumor that is very metastatic, very aggressive, and they migrate a lot. And we take the cells, we put them in a microfluidic chip, we just create a gradient, of chemical gradient, ABS, very simple, very easy to perform, and we put the cells in a hydrogel just to observe how was the migration. Here is the result, but here's the movie. So there was almost no cells moving. This is just dividing, moving a little bit randomly, and so on, but there was not migration. This was a little bit frustrating because this was a very highly metastatic tumor. And we said, okay, what is going on? Oh, yeah, let me think. These cells are epithelial cells. They need to be together. They need to be close, one to the other. Again, let's go back to the histology. And what we created is an spheroid. It's exactly the same number of cells, exactly the same gel, exactly the same conditions, but the results, as you can see here, is completely different. Cells, they migrate toward the gradient, creating a branch going out from the tumor spheroid and starting to proliferate. So this gradient is not only inducing migration, but also proliferation, exactly the same case as before in the glioblastoma. So these changes in the environmental conditions are promoting a more aggressive phenotype in both different tumor types. For sure, as I've mentioned before, there was a necrotic core in the center. It was very important. It was a very clear characteristic just to define this glioblastoma, and we came back again to observe if we were able to reproduce this necrotic core, this ischemic area. We put the cells in a highly dense matter, and we just perfuse the nutrients through the lateral channels. I don't know if it's going to be possible to observe the video. Please, I forgot to put the video here. If you can just click it on it. You will observe something like how the cells, is this video, if you click it over here, this one? Oh, yeah, please. I don't know if it's working. So, as you can see here, these green cells are alive cells and starting to come in red are the dead cells because the lack of oxygen and nutrients. So we are creating an ischemic, self-induced ischemia model just for this tumor. And what is the importance for this, apart from the applications on ictus or also in heart ischemia, is that the cells, they behave completely different when they are in the STERF area in comparison with the normosic area. So we are observing that the cells that are in different environmental conditions, they respond in a different manner to the drugs that we are applying. When we are putting this temozolomide, that is the only drug approved for glioblastoma, in 2D, we observe a 70% of cell death in Petri dishes. However, in the clinical outcome, we observe only 1-2 months of improvement in the overall survival. What we observe in our chips is that we have an increase in the number of cell death, but it's a very tiny increase. So we are just mimicking the in vivo response. We tried, as we observed, that the oxygen was one of the key points we observed, and we checked in the literature which was one of the possible candidates to be used in this system, and we observed that the tirapasamine was one drug that it was activated when there was no oxygen at all. We tried it, and we observed a huge increase in the cell death, but again, we forgot that those cells in the oxygenated border are the ones that are migrating and creating the coming back of the tumor in the patient. So this new drug failed in our chips and also fail in clinical trials. So what we are saying is that we have a very interesting model just to mimic what is going on in vivo. The last question is, we have just changed a little bit the chip. We just want to mimic the metastasis. 90% of the patients die because of metastasis, and this is one of the things that we want to model. Just to use animal models is okay, it's very useful, it's very, very clear, close to the in vivo situations, but it's very hard to monitor. What we are doing here is a microfluidic chip where we can put a 3D culture. On top of it, we can put a 2D culture, for example, breast cancer cells. On top of it, we can put endothelial cells, and we can monitor how the cells are disrupting the endothelial barrier just to go out and to be extravasated on the blood vessel. So these are some data that has been published, and this is just one of the examples that we can just create different chips for different applications, taking into account that we need to know what is the clear and idea, what do you want to mimic in vitro. If you have this clear, this technology is going to allow you to do all the different experiments that you want. Just to finish, I want to thank all the collaborators and all the financial support that we have got in the past, and for sure, as you have seen before the picture of our group, the Applied Mechanics, because Manuel has mentioned, I just want to thank all of them and our collaborators, the support that they have given us. Thank you.